Okay, we're going to get started today. I'm very happy to have Patricio Dandy. Before we get started, I will do an acknowledgement of country. The University of Queensland acknowledges the traditional owners and their custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connection to country. We recognize their valuable contributions to Australian and global society. So Patricio is doing his PhD right now in Paraguay.
Oh, yeah.
Here, but you're from Paraguay.
I'm from Paraguay.
Okay, cool, sweet. Here in the vet school. And so there's a lot of UQ connection with the vet school. People who don't necessarily know, Tatiana is part of CBCS, who's in the vet school. And also Ricardo, he is also part of the vet school, and he led a One Health initiative that a lot of CBCS people were a part of. And there's more. Oh, Nick Clark, who's on like half of my students' PhDs. So there's more of a vet school connection than we often think. So we're happy to have you. So you did your master's at the University of Queensland too, sweet, 2023. And now he's doing his PhD in reproductive biotechnologies.
Yeah.
Cool. All right, I'll hand it over to you.
Awesome.
Thanks.
Yeah. All right. Good morning, everyone. Thank you for being here. I'm Patricio Dandy, as mentioned, and I'm actually from the School of Agriculture.
Oh, sorry.
Don't worry. I'm from the School of Agriculture. That's where I'm actually enrolled. But I'm based in the School of Veterinary Sciences, and all my laboratory and everything is happening there. Anyway, today I'm here to talk about a little bit of what I do in my research, which is the title of today, in vitro production of kangaroo ICSI embryos. And just for the record, this was the first in vitro embryo produced in kangaroos. So very proud of this project. But before talking about the kangaroos and everything, let's talk about what ICSI means if you're not familiar with it. Of course, all of this is thanks to my supervisor, Andres Gambini, and co-supervisor, Steve Johnson. So let's talk about ICSI. So ICSI, means basically intracytoplasmic sperm injection. Intracytoplasmic means that the sperm is injected inside of the egg, and I just need one single sperm to actually be able to produce an embryo, comparing to conventional IVF that I need a lot of sperm. Anyway, now that we know what ICSI means, we can go and start talking about what the marsupials are. So marsupials are in for class of mammals, and there's around 330 species in the world, distributed in the Americas and distributed in Australasia. We have around 100 species in the Americas, where the most common one is the Virginia possum, and we have around 250 species here in Australasia, where the most common ones could be the koala and the kangaroo. So now let's dig in just in Australia. So when I just moved in, I thought, okay, Australia might look kind of like this, full of the marsupials, full of the beautiful biodiversity that they have, but Australia actually look like this, obviously, with a lot of land that is basically just sand, and with a lot of small pouches of green that are mostly related to the coast. In all these pouches is where the majority of the animals, humans, and the environment coexist. But as a student that previously has lived in a house, in a shared house, sharing is not always the best, and it could be a bit of trouble. So that's the same case of the marsupials. The marsupials, these coexist and present threats. This threat includes car crashes, non-domestic species that were introduced, diseases, climate change, deforestation, and of course, human population growth. All of these decrease the marsupial population, and obviously it has the highest extinction rates between all the mammals, with over 40% of species threatened and more than 15 species extinct. And just last week, one of them was extinct as well. That is the Christmas shrew. I don't know if you heard about that, but yeah, a bit sad for the marsupials. So conservation strategies that needs to happen. They are happening. I'm pretty sure some of you are aware of it or working with it. But my type of conservation strategy is related with ART, or assisted reproductive technologies. So that's what I work with. But for those of you who are not familiar with ART, I'm here to tell you a little bit more about that. So ART are technologies that help us to produce embryos or produce babies, basically, in a lab facility. So on one hand, we have artificial insemination, where we deposit the semen inside of the uterus of the female, in vitro maturation or IVM, that basically I can collect from a dead or from a live animal and oocyte and put that to mature to become a mature egg. That mature egg can be used for ICSI, that you're already familiar with that term, basically to introduce one sperm inside of the egg, and then we have the conventional IVF, that we have a mature egg, and that mature egg is fertilized with a pool of sperm. We let them do their thing. Subsequently, other type of ART is cryopreservation that basically help us to preserve the samples, whether sperm or eggs, and we can use it in the future whenever we want. So all of these ART have been implemented in multiple species. That have been implemented in domestic species as horses, dogs, cats. Actually, in our lab in the vet science school, we have an ICSI program. So basically, everywhere we're producing embryos, like horse embryos. IVF has been used a lot in the cattle production. Most of the food that we eat could be from IVF. Human IVF has been helped a lot in infertility. Today, over 80 million of babies have been born after these technologies. And of course, it has been helped a lot in conservation of a lot of wildlife species. When we talk about marsupial assisted reproductive technologies, we can only talk about the production of embryo, the successful production of embryos in koalas after artificial insemination. So only with all the marsupials, just koalas have been born after artificial insemination. And it has been tried in the wallaby, but sadly there has been not babies after this. So me and my friend the kangaroo, we think, what should we do, what we can do to try to help not just the kangaroos, but all the other marsupials. And what he thinks is like, okay, in Australia it's basically 2 kangaroos for 1 human. This is, yes, distributed in 3 species of kangaroos, but today I'm going to talk about the Eastern grey kangaroo, that is a species where specifically I'm working with. So we think that he can build a model to other marsupial assisted reproductive technologies. It's not about the kangaroos only, it's about what we can start building for other ones, for the most endangered ones. And the reason why I choose as a model the kangaroo is because 2 things. The unique reproductive biology, that they mostly share this with all the other marsupials, but particularly in the Eastern grey is their overpopulation. So let's dig in a little bit more in these 2 things. So the unique reproductive biology, if you're not very familiar with an oocyte, that's how a mature oocyte looks like in a mammal. Probably in some of the ladies, of course, it's going to look like this. So we have one layer to protect the egg that is called the zona pellucida. This zona pellucida is what the sperm has to penetrate during normal fertilization. In the case of the marsupials, they have 2 extra layers that are formed after the ovulation of the egg. So they are formed in the oviduct or in the uterus, and there's actually no knowledge of how this affects the actual fertilization, or how do we need or not need these extra layers for the production of embryos in vitro. Another thing that we have is, this is how a normal embryo looks like in mammals. So this known as a bilaminar blastocyst. Basically, it has an inner cell mass. In between these 2 groups of cells, they are going to differentiate in what is going to be the placenta in the future, or what is going to be the actual baby. And in the case of the marsupials, they have something called a fluoroblast. From this fluoroblast is where they're going to have starting to form their placenta and also the actual baby. As you can see, all the extra layers is still there for the formation of the embryo. So as I mentioned before, we still don't know how important is or not these extra layers in the production of embryos in vitro. And lastly but not least, in the case of the kangaroo, they have apical jaws, so they are very tiny little jelly things that has very, very well developed forelimbs and a sucking mouth to go from the pouch to the pouch and basically start sucking the teeth and develop after these 36 days of gestation. Now that we know that, we can talk about the overpopulation. So kangaroos are known because we can see them everywhere, but the things that they are affecting a little bit the actual agribusiness, or they are actually also being used for the production of meat. So that gives me in 2 separate ways. Animals are being killed because they are affecting the production, or because we are going to use it for food, as well as car accidents. Overpopulation means there are more kangaroos going to the road and they die. But why am I in the conservation talking about dead animals? Well, if they die, of course, I have a dead animal. But what I can do is gamete rescue. I can recover the cells of those animals who pass away, or animals that assisted to wildlife hospitals. I can collect the eggs, I can collect the sperm, and I can produce the embryos with them. I don't need them alive. I just need the cells alive. So what I do with that, I collect these cells and I produce in vitro. I mature these eggs in vitro, and then I do ICSI to actually try to produce these embryos. So that was my bit long introduction. So now we can talk about the aims. The aims of this study was to assess post-mortem Easter-grade kangaroo oocyte recovery and morphometrics and in vitro maturation rates in media developed for domestic species, and also to explore embryo developed, including cell number and DNA fragmentation levels following the in vitro maturation and the ICSI. How did I do this? So whenever an animal died, I was able to access the samples collected around 2 hours post-mortem. So I went there, I was lucky to have 2 ovaries per female. So what I did is started to puncture these ovaries and also slide it a little bit with a scalpel blade to release the eggs. So I was able to have the eggs like this, and then under a stereoscope, I was able to start to search for it. And then what I did is to evaluate the oocyte diameter. So we want to know the size of the egg and also the thickness of the zona pellucida. And these eggs, all the ones that I found, were without these extra layers that I mentioned before, because of the reason why those extra layers form after the ovulation in the oviduct. So subsequently, I collected my eggs, and they were like that image with zona pellucida cover. That's basically the first cover of the egg. And I know that they were immature oocytes because they have the germinal vesicle. That tiny dot in the middle and this cover, there is a zona pellucida. That means these eggs are immature and that I can put them to mature because they need all of these to actually mature in vitro. So what I did is culture them in 35.5. That's what the literature says, there's the temperature of the marsupials. And I knew they are mature because they don't have anymore the zona pellucida, and then instituted the polar body. And that basically means these eggs are mature, and I was able to see it around 30 hours after the in vitro maturation. So subsequently, what I did is my ICSI experiments. I wanted to start the injection of the sperm inside of that. So after I have like 2 experimental groups, my mature eggs that I did the injection of the sperm, and my immature eggs that we use this control group because an immature egg are not supposed to develop into an embryo. So basically we wanted to double check everything. And what I did is use chilled sperm, which basically means I collect the sperm from a dead kangaroo as well, and I chill it. So basically both sperm and egg were from dead animals. After that, I cultured them for around 4 to 5 days, and I was able to assess cleavage after 48 hours. So cleavage, if you're not familiar with the term, basically means when the cells division start to happen. So that's when I start to see 2, 4, 8, vice versa. Then what I did is immunofluorescence to evaluate the DNA fragmentation, so the cells that are starting to produce in these embryos. So I used my first antibody that is known as gamma H2AX. This basically talks to you about like DNA fragmentations levels, but also talks about a little bit the repair of the DNA. So when the cells start to divide. And I mounted with something called DAPI. DAPI basically shows me the cell number. So basically then we did confocal microscopy, and I was able to compare and contrast the numbers of cells and the percentage of DNA fragmentation levels. We know the methodology now, and so we're ready for the results. So basically, I was able to recover 139 oocytes from these 11 females, and that's how the beautiful immature oocyte looks like, covered by this zona pellucida. And then I was able to assess the diameter. Basically, I was able to evaluate that they measure around 181 of diameter and zona pellucida of 7.5, which is basically very, very thin. But again, this itself doesn't look amazing. But if I put next to, for example, a cattle egg, we can see the big difference. The oocyte diameter in the cattle is a bit smaller, it's around 152, and the zona pellucida thickness is 13.5, which is basically double the size of the zona pellucida of the kangaroos. We don't know why. We don't know if we still need those extra layers that the kangaroos forms during the oviduct and everything, but it's very nice to know these kind of differences. Subsequently, I did my IBM rate. So I put 106 oocytes recovered, and I was able to have M2 means mid phase 2, that means a mature egg. So basically 34 were able to mature, 37 means phase 1 is immature, so they are the ones that did not mature, and then 35 is generated. But if I do, or if I'm working, for example, in a conventional IVF place, we won't count the generic ones. We're just gonna count the mature and the immature. So we can say that I have around a 47.9% of maturation rate, and we can see that they are mature because they extruded the polar body. So then I did my ICSI and my cleavage assessment. So from 11 eggs that were injected, 10 of them actually cleaved. That means I have almost a 91% of cleavage of embryos that we injected and started to develop into actual very early babies. We injected also our terminal vesicles, that was our immature eggs to control. Then we have zero cleavage, that's what we were expecting, so we were happy with that. But then what we were able to see is that they develop, or they were arrested around 600% of them, between 4 to 8 cells. Of course, this is not great, but this is a beginning. And then we were able to see that they were arrested around 40% of them, between 10 to 14 cells, which again is a beginning, and we were very happy with this. After that, what I did is like from the 10 embryos that I was able to inject, I evaluated the DNA fragmentation levels and the cells numbers. So in my left, right, you can see DAPI, all the kind of like blue dots, those are all the cells. And then the red dots is the one that you can start to see that they are like DNA fragmentation levels. And here we have the merged image that we can see that, for example, this cell has a DNA fragmentation, this one has a DNA fragmentation as well. So that's how we started to have a DNA fragmentation index, and it was around 0 to 38%. It's a bit high, but again, it's kind of like the first time that we were trying everything, and it's a result anyway. So as a conclusion, we were able to recover viable oocytes from the cis animals, so that's a win. Oocytes can successfully be mature in vitro and develop into early embryos following ICSI using media adapted for domestic species. But of course, we need to still make some touches to make it more suitable for these marsupials. And this finding represents a significant advance in marsupial in vitro embryo production, but of course, further investigation is needed, not just for the kangaroos, but also for all the species. And that's it. Thank you so much.
That was excellent. That was beautiful slides. I haven't taken biology since high school. And that was really a good refresher. We have plenty of time for questions, and there may be questions online too, but we'll start with those in the room.
Yeah, so I wanted to know, have you been able to reintroduce the eggs like after they're fecundated to the kangaroos, or?
No, I would love to do that one day, but of course, there's a lot of not issues, but like steps that we need to go before doing that, and all of that is especially animal ethics, and also the reason that it's not a protocol to actually do that. According to literature, in kangaroos should be easy to do it because they are kind of like similar to the sheep. So in sheep, you can easily do just the injection of the sperm and things like that. So they kind of like say they're similar to that, but of course, no one actually did it. But yeah, it will be great if one day we can actually transfer to an animal and say that we can actually produce an embryo or a baby this way. I know trials are starting to be done in a very tiny species like donards, for example. But yeah, in big species like this, not yet.
I know like kangaroos are embryonic diaploids, something like that. They can have like up to 3 stages of
Yes.
different. Does that affect like how the embryo is being treated or how it grows up, or?
That's actually a great question, and I used to have that in my slide in the special biology of the animals, but I thought it was going to be like too much to talk today. So it's called diapause. That means that these animals can have like more than one baby. And specifically, the Easter grade, it's facultative. It's not that always happens, but actually it did not affect anything. Like they're pretty cool animals that they can have, for example, white babies out of the pouch and one mammary gland is be producing one type of milk. The other one can be a baby on food, and this other glandular mouth produce another type of milk. And the embryos that are in the diapause, it's crazy because it's not being described yet how they actually are there. Like they are literally just floating around the uterus till the sucking stimulus finish, and that's when they start to develop or like implant onto the uterine wall and it starts to develop. But yeah, like from my like literature review and everything, there's no reports yet saying, okay, this is what happens or not. They are basically just floating there, and at some point when the sucking stimulus finish, they start to develop.
Thank you.
What was it?
Right.
What does the DNA fragmentation mean?
So the DNA fragmentation is basically to tell us if the cells are dying or not. And basically, we see a cell that is in there, but it's not good or is not alive, or is it basically just dying? So that's why it's a bit good to know, and that helps us, for example, to know if our media is doing the right thing or not, if our cells are trying to die earlier than what we're expecting or not, or if the whole process itself is working or not. This is very, very high. Usually, if I'm not wrong, it should be less than 15, for example, in cattle and things like that. And the cells that we were able to find, they were like only around 14. When we talk about around 200 or more, usually like in domestic species. So yeah, it's basically just to let us know if the DNA of that cell is being damaged or not.
Yeah, thanks. Could you talk a bit more about how you samples or how you extracted or chose the animals to extract the gametes from? So it's just opportunistic roadkills, or why didn't you? Could you have used like the home pine kangaroos or?
Say it again, the last part, please.
Could you have used kangaroos in captivity, or did you?
We could have definitely, we could definitely use kangaroos in captivity for like the male side, for example, like the semen collection. If I'm not wrong, there's no reports of collection from live animals kangaroos. I know that in koalas, there's a reports of like artificial vagina and everything. My co-supervisor is the one who developed that. But I'm not sure if there's like an artificial vagina for kangaroos. If I'm not wrong, they do electric ejaculation, and that has been reported. But it's just tricky, needs animal ethics and everything. And like using the dead animals as something that we can start developing, or like just test if what we're doing, like the in vitro reproduction works or not. Using these animals that are dying without touching animals alive yet before developing all of this, we believe it's something bigger and great. To implement in animals that are alive once we have everything basically set up. Most of these animals are often being called to mitigation permits in private properties. So they did the cooling, and I went there. I was able to get the samples that were fresh. So basically, I'll just open the body, collect the ovaries from the females, or in the case of the male, the testes. But at the same time, we work in partnership with our CBCS. So animals that, for example, they've been hurt, or for some reason they need to go through euthanasia. They call me, and I go there and I collect the samples from the animals.
Well, what was the spatial spread of samples? Thinking of genetic diversity. So you got them all just about you can switch them over.
Yeah, that's a very
That's genetically
yeah.
That's
a very good point. Definitely genetic diversity is something very important when we are talking about the production of embryos of babies in every species. As I mentioned, we're just trying to set up this protocols. So far we're not thinking too much about the actual genetic diversity, more about saying it works or not. But definitely if this goes farther, we definitely need to take care into consideration, like which male we're using, if it was the daughter, if it was the brother, sister, mom, all things like this. But yeah, so far we're a bit far from that.
Like not specifically with kangaroos, but do you know if like there's instance that they use it for that, for like genetic pool diversity or something? Or
in other species, it definitely has been done. Most mammals has been used like genetic recovery. It is still being done. For example, like we have working with horses that we collect the semen from the epididymis of these horses that they died ages ago, and we're still using these sperms to produce more babies. That we are aware of the genetics of these animals. But for wildlife, I'll lie to you if I say that I know a lot of that, but definitely I know from domestic species, it has been done. It's still been people do it. We are doing it in our lab.
What are the next steps? What would you play around with in order to improve the results?
So the next steps is continue to have more numbers, and we are doing some tricks that are not published in this. So we're adding other hormones in the media to see if that increase or not the development of the embryos. And another step is also like working on other species. For example, I'm working also with the koalas. So that hopefully is going to be a paper at the end of the year or the beginning of next year. But yeah, basically, our future with this is like just to say it works or it can work or not. But for my PhD, I don't think I'm gonna have enough time to go much further with it. But yeah, I would love to like actually be able to access to much more samples so we can actually try much more things. Because I probably get like one animal per month. And with that, I cannot have a lot of numbers. And we need numbers to actually develop more, all the protocols and everything. But yeah, I would love to like have access to more, to work more and develop more and have better results, of course.
Okay.
You mentioned the surrounding walls and that
Yes.
there are three pellucidar,
Yeah.
pellucidar, and then the thickness of the cows. Why is that? Out of curiosity, is there some like, is it just out of curiosity, or is there something that?
So the sonopellucida actually is the part that, well, first of all the sperm is sonopellucida specific. So the sperm of each species is specific to the sonopellucida.
Mhm.
And the sonopellucida is the one that protects the embryo. Then it's the one that after the what's the name? The fertilization happens. And of course, happen the blocks of the other sperms that are around. Is the one that is going to keep the embryo inside, is the one that is going to protect the embryo. So this thickness is the one that basically, for example, in cattle, helps to protect the embryo or the development of it.
Yeah.
And it has been guessed that in marsupial, these other extra layers are the ones that actually help in this way. Those extra layers that forms during the descendants of the oocyte from the ovary to the oviduct in the uterus. Because then they add this extra that protects, for example, even like the sperm. So in cattle, you have what's the name? The tubular cells and the sonopellucida and all of that helps to the sperm, to the selection, the natural selection.
Mhm.
And in marsupials, it's been told that these other extra layers also helps with the selection of the sperm, to say which one is the best or not.
Filters.
Exactly.
Yeah.
So they are filters. It's also not known if they actually provide some type of nutrients inside of the egg. Because like one, for example, is one of the layers. There is a mucoid that's glucoproteins. The other one is like more of protein based one, kind of like similar in evolution to what is going to be the eggshell.
Mhm.
So that's why it's yeah, it's just things that are not known yet.
Yeah.
But somehow, of course, they are needed for the normal development of the animals or the eggs inside of the uterus. And something that we cannot replicate in vitro because we just collect it from the ovaries and they just have one layer, not the other extra two.
Oh, so that's why the interest is like, what's the purpose of those so that you can
Exactly.
replicate it? I
Yes,
see.
yes. If it's needed or not,
Yeah,
could be that the reason why we're not developing further because they need that.
yeah.
So yeah, it's.
And you said it's
question
14
marks.
days, is as far as you got.
No, 14 cells.
14 cells.
Yes, then just 4 to 5 days
Okay.
is the one that we develop it further. And basically, you can go up to 8, 9 days for the development of in vitro embryos in normal domestic species, I'm saying. But in our case, we stop it in between those 4 to 5, because according to literature, that in normal animals, like live animals, that they cool and they evaluate the collection of the actual embryos, at that stage, they should have like over 50 cells. We did not reach that. And by the time we assessed those cells, of course, that we're not moving farther anymore. So there was no point of living there without like growing. Because the development should be faster. Like after you have 4, 8, they just multiply it a lot. And that was not happening. So that's why we basically stopped at those 4 or 5 days.
Okay. Thank you.
Thank you very much.
I have one quick question. Why is it so different? Like, is it just no one did it for marsupials yet? And is the reason because the eggs are more complicated? Or not more complicated, but just substantially different? Or was it only a matter of time? We just needed to go through it? Like, was there any like major breakthrough besides the weird egg?
Yeah.
Yeah,
How will I? If I say that, I just say is a species like is a group of species that people are not taking into consideration a lot
Yeah.
to work with. And one of the big reasons are because there are not domestic counterparts of them. Like if we work with I don't know, tigers, we have the cat. If we work with wolves, we have the dog. If we have with zebras, we have the horse. There's no domestic marsupial that has been worked before. So that's why we need to try to find a model species, and that's why we're suggesting the kangaroo because of the accessibility of the samples. There are a lot of, like, for example, research being working in Donards. I think one of the biggest breakthrough in marsupial in vitro and everything is through Donards, and also there is a lot of old work on Tamar wallabies, but most of them are just recovery, not the actual production of these embryos in vitro.
You talked about the difference between, I forget the name, ICSI
Yeah,
and
IVF.
IVF,
Yes.
but then I couldn't really tell what the difference was. Can you do it again?
Yeah, I can go back there.
You teach this, yeah? You
Yeah.
teach this. These are great slides. Good, you're a good teacher.
Thank you. So, ICSI or intracytoplasmic sperm injection, how it says, basically we implant inside of the egg only one sperm. That sperm can be motile, that sperm can be dead, that sperm can be not moving at all. We don't need it to move it. Like literally, when we are collecting this sperm, what we do is like kill that sperm, and we need it in model so we can suck it with this tiny needle. This tiny needle that is known as the ICSI needle. So then we inject it inside of the egg. We aspirate a little bit of what is inside, and then we inject it inside. So that's basically intracytoplasmic sperm injection.
So
And
it's a dead sperm.
is basically yes. What we do is break the membrane to release basically the sperm inside, and we don't want it to be moving inside of the egg as well. As long as the DNA is intact, we're happy with that. Like all the things that I'm doing is working with freeze drying. So basically with freeze drying, we dehydrate the sperm and it's just literally a powder. Like everything is kind of dead because it's not moving. But the nucleus remains intact. And there has been reports in horses, in mice and everything that babies has been born and they are amazing after the freeze drying of the sperm, after ICSI, and they are running somewhere around the world. So
freeze dried is different than the regular Cairo. Because like for humans, even we have the tanks for frozen eggs. And,
yeah,
but that's wet, not freeze dried. That's freeze.
freeze is cryopreservation. So that's basically that one.
So you dehydrate it. Yeah.
Yeah, we dehydrate.
That's just powdered sperm.
Yeah, basically.
But the egg, so there's a reason why you have to cut the membrane open is because normally a live sperm, when it comes through the wall of the egg, it like that process of going through kind of gets it ready to release the DNA.
Yes.
But if you drop a live sperm into the middle of an egg, it's not gonna know that.
Yeah.
It's just gonna go moving around.
Yeah.
And that's not what you want.
Yeah.
All right, and so the IVF, then you put the bunch of live sperm next to the egg, you let one get in,
Yes.
or you hope one gets in.
Mhm.
Sweet, that's cool. So only horses do freeze dried sperm so far?
No, there's plenty of species. Actually, in marsupials, I think it was the Donard that has been reported that they have been done freeze drying already. They haven't done ICSI with that freeze dried sperm. But
Do they do ICSI for humans? For
yeah, totally.
okay, so that's one of the methods for
Human IVF.
so because they complain about low sperm quality,
Yes.
especially in men of the young generation. It's a big problem apparently.
Yeah.
According to the news.
Yeah.
But dead sperm are just as good. We learning.
Yeah. As long as the DNA is intact,
Cool.
we can use it.
What were you saying about aspirating to the part on the left? You're
Yeah.
taking some out to compensate?
Yeah, so when we aspirate, what we want to do is kind of like break a little bit of the membrane, because if we just put it in, what happens is this membrane that we can see here, it's not broken. And we usually put it like outside. So if we put it outside, it's not gonna grow. So what we need to do is aspirate a little bit, kind of like break that membrane to the sperm actually get in. And yeah, that's the reason why we need to aspirate a little bit before putting in.
Why do ICSI and not IVF, or how did you choose?
So we choose because what's the name? The benefits of being needed to use only one egg, one sperm. So we have one egg, one sperm, we put it in, done. With IVF, we need to put a pool of sperm, and we need to see if the sperm is actually able to do it itself or not. And there's no reports of IVF in marsupials, so that means that also there is no development of the media or all the things that we need to put here to actually allow them to fertilize in vitro. And it's easier if you just put one in than with culturing. Does that make sense?
Sorry, you're saying that IVF has been done and not been successful, or it's just not been done?
From my research, I don't think there has been done any IVF in marsupials yet. I might be wrong, but I'm pretty sure there's no IVF like the conventional IVF.
So you're saying it's not established in terms of media protocol?
Yes, yes,
Easier to know.
yes, 100%. There's no establishment of any protocols of in vitro in marsupials. At least not like very, very established.
Do you think you would try that?
We are trying to use it with domestic species media at this point. My co-supervisor definitely wanted me to do it with koalas. He really wanted me to do it with koalas because something that is very cool with the koalas is they have 11 head morphotypes. So they have,
What was that?
the koala sperm has 11 head morphotypes.
What's a head morphotype?
So a head morphotype is like this. So in normal.
Oh, the head of the
mammals,
sperm
yes,
is different.
yes. So
Different shapes?
they have different shapes, it's crazy. Like some of them are like this, like they're like a question mark. And there's literally no literature saying like how that thing that is like this swims like this and then somehow gets in or laid down onto the egg or something like that. So that's one my co-supervisor definitely wants me to evaluate this.
It's a good idea.
Yeah. So we will see.
But so like roos only have one head morphotype?
The roost has, yes, one head morphotype, but it's pretty cool as well with them, because like they start with the head like a T form. And through the time that they go through the epididymis, which is kind of like the maturation process of the sperm, they go like this, and then like this, and then like this. So the most mature sperm is the one that forward, sort of like this, it's like an arrow shape.
Yeah, how long can the animals be dead for in terms of extracting the eggs and sperm?
That's a very, very good question. In the case of the females, we want them as fresh as possible, because in the case of the ovaries, we want it kind of warm or at room temperature. We don't want much to them to go cold. In most species, they can happen like that. We can have it cold, and they can still develop further. But since there's no reports of how long we can have it cold or not, we just want to do it ASAP. So my animals that I mentioned before, they kind of die, and after 2 hours, I go collect it, keep the samples on a ski box, go to the lab and collect it as soon as possible. With the males, I have been working on the chilling process. And I mentioned before, the sperm that we use for 72 hours post chilling. So that so far is still being alive after 72 hours. But that's again after the collection. We want them basically out of the body as soon as possible. So our kangaroo die, we go to our SPCA, we cut the testicles. And yes, the testicles we can put it inside of the fridge. But in the case of the ovaries, we go as soon as they die, we cut it and we go and work with them. And definitely we don't want it to like warm up or being like in the heat or something like that. We wanted to be in a kind of like controlled environment that we can know how fresh or fat they were before we cooked.
I'm just curious for you, how did you get into this specifically?
So I got in there because of my co-supervisor, Steve Johnson. So basically, what was happening is like there's a lot of koalas arriving to our hospitals dying. So my main project is more towards the koala part. Well, hospitals in general. But we started with the koalas, then we saw a window where we can include also the Eastern grey kangaroo. And that's basically the reason why I started to get that. But I'm international, I'm from small country, South America. I never thought I would be working the way, so I'm very happy to be here.
Yeah, just you mentioned this overpopulation.
Yes.
So are they, is that referring to kangaroos or humans?
Kangaroos. Well, humans are being overpopulated too.
So, but are they overpopulated?
Yes,
I
yes.
thought they were just.
Yeah, kangaroos
I
are
think there's a
definitely.
danger of population that becomes used as a pest species.
Yes,
So
yes,
I'd happily say kangaroos are overpopulated,
yeah.
but I'm not sure, is there, have you? Just support that,
That they are overpopulated?
I don't know what.
They call them at times, like here in Australia, they call them because they are like overgrazing and
Yeah,
like
yeah, and
overpopulated.
that's. So is it just overpopulated from the point of view of agribusiness?
Yeah,
Because
exactly.
it's a real
Yeah.
environment here anyway, so, yeah, just. Like, it's not that.
Yeah, like they very much food and things like that, that they start like dying on their own, cause like the other ones kind of reach like, so that's why they. Oh, I think. That's what I hear.
Is that natural, so?
Basically, yeah.
They like.
And the reason why there is a lot of spoil is because of this specific strategy that they have, that they can basically breed all the time. They can have one embryo of the uterus, one tiny baby, one kid, and the other one fruit. So they're breeding all the time.
Yeah, but they can also, they have diet boss, so they can
Yeah.
both breed with conditions.
Exactly.
They
Yeah,
involve-
they're so cool. So if you want to scan it, you have the ICSI video there to understand like how the whole process is. And see how we kill the sperm injected in.
Okay. I think that's enough. We can say thank you. Very interesting. I learned a lot. Thanks. Appreciate it.